It's an honor to introduce Kirsten Tusscher, not only because she's a highly accomplished theoretician, but she's also a faithful and active member of our society, also just organized the symposium this afternoon. She studied in Utrecht, where she did her PhD, and she worked on cardiac arrhythmias, and she continued that in Oslo during a postdoc. But then when she came back to Utrecht in the Netherlands, she started her own group and started to work on an impressive program in Evo-devo, touching upon really almost every relevant concept in Evo-devo. And very importantly, she's addressing the big gap that there is between gene regulation and morphogenesis. So we are very happy to have her here, and please start your talk.
Well, thanks for these kind words and for the invitation. Let's hope I can live up to these quite high expectations. So what I would like to talk about today is iterative body plan patterning in animals and plants, and hence the subtitle of mice and plants, but perhaps it would have been more appropriate to speak of reindeer and trees. So what I would like to do today in this talk is to demonstrate to you the usefulness of computational models in Evo-devo research. And I'm guessing that most of you are now aware of the use of these models when it comes to understanding how particular processes work or how they evolve. But what I want to stress in this talk is also that they're very useful to understanding why patterning mechanisms work the way they work, or why they have evolved down to that particular path. And furthermore, I would like to show you some intriguing parallels between animal and plant axial patterning mechanisms. So what I want to do, let's see if this light is, oh, it's not so clear. Well, what I want to do is I want to start with a specific example of something that I worked on in plant root patterning, and then show you some parallels to animal body axis extension. From there on, move to axial segmentation and its evolution, and then come full circle back to plant root patterning. Well, I guess most of you know that animals and plants have diverged evolutionary a long, long time ago, approximately 1.6 billion years. And even though perhaps now ideas are shifting that the common ancestor wasn't single-celled but might have actually been colonial, it is quite clear that all the complex patterning that is going on in development is really not homologous between animals and plants. So they really represent sort of independent evolutionary experiments, so you like. And I think that most developmental biologists are brought up by the idea that, well, there's fundamental differences between plant and animal development. For example, in animals, cell movements and cell adhesion, driving that is very important if you think about gastrulation, neurulation, migration of cells. And in contrast, in plants have cell walls, so these cells are glued together, they're sort of stuck with the neighbors that they have, and they can only change shape by regulating where there is growth and in which direction. Other differences are, for example, that in animals, most development is basically pre-embryonic, and after that, okay, there are some rate differences, because otherwise we would all have this kind of head. But other than that, well, we don't get more legs or more arms, etc. And in contrast, well, these embryonic plants are not very interesting, and most body plan formation occurs later on in life. And then a final important distinction is that in animals, most developmental patterning is kind of blueprint-like. I mean, even if we would scale these dogs to a similar size, then they have the same number of legs at relative the same positions, etc. And in contrast, in plants, environmental conditions play a huge role, and there's much variation. But there's also interesting parallels, and that's what I want to talk about today. And one famous example is, for example, the homeodomain genes, and that you can use them in a sort of combinatorial manner to specify a position or to specify an identity, such as, for example, the Hox genes do along the anterior-posterior axis, but also the ABC genes do in specifying which floral organ to form. So this is as a context. So let's start with this specific example that I promised you. And this is about plant root development. Well, as I said, plant roots are encased in cell walls. So if you want to have growth at a certain location, what you need is a meristem. And those consist of slowly dividing cells that keep replenishing themselves. Then slightly above that we have a zone of so-called transit amplifying cells that divide really fast, that are really the machine driving the growth. And then after that, cells expand and differentiate and they get their final functional roles. And to define where stem cells are and when they start expanding and differentiating, plants use a so-called auxin gradient. So auxin is one of the main hormones driving plant developmental patterning. But plants also have to do different things. As I said, it's very important that they can adapt to environmental conditions. And for example, if a plant topples over, then the top part has to orient away and the root part has to orient towards gravity. And the way to achieve this is to bend, and in order to bend, the top part has to grow faster than the bottom part, and this arises through an asymmetry in the auxin pattern. And because auxin gets elevated at this side, here there's a break on the expansion of cells and therefore it can bend. And then finally, as I said, most plant development is post-embryonic and then they make all these new lateral organs. And because, well, there can't be any migration to sort of provide new material there, so formation of a new organ entails the formation of a new meristem and hence the formation of a new auxin maximum. So basically, this hormone auxin seems to do all these different things. So doesn't that somehow conflict? If we want to have this stable developmental zonation, we need this auxin gradient. But if we want to, in a fast and transient manner, adapt to the fact that we've just toppled over, we need this auxin gradient. That seems to conflict with one another, right? Doesn't this ruin that? Or put differently, how can a single signal determine both what to do, to divide or expand or differentiate, or to determine for a single process to do it faster or one side than the other? How can you encode that information? So the people from the lab of Ben Scheres, formerly in Utrecht, now in Wageningen, they for a long time realized that there is this important family of transcription factors called plethora that seem to be induced by auxin, but it's not exactly clear how. And they, well, it's a highly redundant family, but if you would sort of overlay these pictures, you would see that they have a similar kind of gradient with a maximum here and then tapering off towards the shoot, as does auxin. And they also found out that it's actually auxin itself that directly dictates these rates, for example, if you want to bend, but that it's plethoras that dictate the zones. But well, I mean, if auxin induces plethoras, who cares? Well, the thing was that there was something with this auxin-plethora relationship that they couldn't exactly figure out. So we decided to combine forces and to do both experiments and modeling. So how does a model like that look like? Well, you see, it's a little bit crude because there's not such a nice tip. In later models we did that, but here it's still a little bit squarish. But what we do is we put in, for example, we have epidermal outer layers and a cortex and an endodermis and vascular layers. We have the stem cell region, we have sort of a columella and a root cap, and we put in that there is this stem cell niche and that we have these different zones. So that's the first layer, having this sort of anatomy, this layout. Then on top of that, well, we want to look at things that are driven by auxin patterns, so we somehow have to simulate this auxin dynamics. So we put into these models that within cells there is production and degradation of auxin, there is some diffusion processes, but then there's also a lot of active transport. We have auxin passively coming into cells, actively coming into cells, but the main important thing is that when auxin is exported from a cell to the cell wall such that it can propagate to a new cell, that is being done by so-called pin proteins. And these are not on all sides of the membrane, but for example in the outer tissue layers they're mostly upward, and in the boundary they are laterally, and then in the vasculature they're pointing downward. And this gives rise to what has been called a sort of reverse fountain. So here the transport is down, then here it's in all directions, and here it's up and inward again. And this gives rise to a sort of recircling of auxin that generates a very robust auxin peak here, and then a tapering of gradients. Then, well, this is the part that we have to figure out, but somehow then there should arise a plethora gradient, but well, this is what we're going to figure out. And then plethora and auxin together should dictate some growth dynamics. So this is a sort of pseudocode of how that works in a model, and basically what we put in, well, if my plethora levels are very high, I'm a stem cell, so I sit tight. If it's a little bit less high, I'm a dividing cell, and otherwise I will start expanding and differentiating. And then these processes in their rates, they depend on auxin. And because cells have a resolution of these grid points, I can simply grow them by every now and then adding a row of grid points. And at a certain size they divide, and we assume that the daughter cells inherit this polarity pattern of these auxin transporters. We assume that if there is like cytoplasmic growth prior to division, that things dilute, but that as soon as they start expanding, most growth is in the cytoplasm and there's no dilution. And we assume that at a certain point they start differentiating, and we just indicate that with a different color. So this is sort of how these models broadly look like. And then, well, the first naive thought was, of course, that everything is simple and trivial. Auxin induces plethora transcription, so the plethora gradient is just a readout of this auxin gradient. And this turned out not to be the case. If in the experiments we would throw a lot of auxin on the plants, then only if we throw a lot on it and and we leave it there for a long, long time, you get expansion of the plethora transcription domain. So this tells us that you need high levels of auxin for a prolonged period to induce auxin, to induce plethoras. And the only region where that normally is the case is in the tip of the root. So basically what you get is you get very localized plethora transcription. And if that transcription would directly translate into a protein distribution pattern, I wouldn't have a gradient at all. But then what they found is that it seems that these plethora proteins move through plasmodesmata from cell to cell. So first we did that in the model, and we're like, nothing happens. And we thought, oh yeah, of course, this transport is very slow. So what it implies is that it should be a very stable protein, because, well, if you don't live very long and you move very slowly, you don't get very far, right? So this protein had to be stable. And then, in hindsight, we could find that back in our experiments. But then there's another interesting implication, because if your protein is very stable, it also means that even though cells through division leave this zone where plethora is being transcribed, they still have this protein and it's decaying very slowly. So the level is sort of reflecting how long ago it was that you left this zone. So you have basically two processes contributing to this gradient, both the cell-to-cell movement and this degradation, this slow degradation. Okay, but still, who cares? Well, we started out by wanting to resolve this discrepancy between development and adaptation. So let's bring the adaptation back in. Let's simulate gravitropism. So what is known from gravitropism is that the pins that are in the tip of the root that normally point in all directions, all of a sudden only point downward. So now this direction is downward, the pins are pointing there, and indeed you see all auxin is being pumped in this direction, and you get an asymmetric auxin profile. But because there is this long time scale in influencing plethora transcription remains almost perfectly symmetric. And because there's another long time scale in plethora movement and in plethora degradation, the protein gradient remains symmetric. And the only thing that happens is that because here in the expansion zone we have high auxin levels, this is suboptimal, expansion rates decrease, these cells don't become long so fast, so this zone shrinks and the differentiation zone moves in. And admittedly it doesn't look so nice because in this model there is not yet actual bending. We're working on that right now. But you see that there is a very nice agreement with experiments. Auxin is asymmetric, plethora is symmetric, and if we look at the root hairs that are the first signs of differentiation, they are closer here than they are there. And even the cell number difference fits very well. So what this tells us is that basically there is a division of labor going on. So auxin directly influences the rates and thereby allows it to do the fast things, the environmental adaptation. But by working through these plethauras that have their own way of making a gradient in a very slow manner, they ensure that this developmental process, for example, this boundary between meristem and elongation zone, that is stably maintained. And the nice thing about models is that you can do crazy things that you cannot do in experiments. So you could do in this model, and we did that. Let's assume that plethauras are a readout of auxin. Then these two patterns also become asymmetric, and this boundary becomes asymmetric. And then later on, when the root has reached gravity again, this doesn't get restored anymore, and you sort of screw up your entire nice organization of differentiation. So this is what I meant by showing that you can disentangle both how it works, but also why it makes sense to do it in this particular manner. But when I started out about these many roles of auxin, there was yet another thing, namely this making of new meristems. So what we did then is simply take exactly the same model, but only start with one tiny row of cells. And these cells basically correspond to cells that have obtained the potential to form new lateral roots. I will come back to how that occurs later. And see whether they can actually bootstrap themselves into forming a new meristem and then a new lateral root. Well, and first that sort of failed miserably. Because, well, simply couldn't reach high enough plethora levels to actually define a meristem. But what my experimental collaborators have been working on is that these plethora genes, they have zillions of targets. But if we focus on the interesting one, there's many targets that are involved in auxin, either stimulating auxin biosynthesis, inhibiting auxin degradation, promoting auxin transport. So then, as a theoretician, you immediately think, yeah, I have a nice positive feedback loop, and that gives me this essential bootstrapping potential. And indeed, if we put that in the model, we get a nice elevation of auxin, we get the plethauras, well, this is the tip of the root, so here's the auxin tapering off, we get plethauras going, and we see a stem cell niche with a meristem. But we also see, well, this doesn't really fit very nicely. So basically what is happening here is that things are working too well. Now I have this positive feedback loop, and there's no thing limiting it. Each cell can make its new auxin, and each cell can transcribe its own plethora genes. So what's the break in the system? So the first thing that we realized is that in this simulation we assumed that this convergence point of this auxin transport machinery, that it could move along with the meristem. But then, talking again to our experimental collaborators, they said, well, there's only very rare mutations that this can actually happen, but in principle all growth is above it and this focus point remains fixed. So when we put that in the model, you immediately see that because you keep focusing the auxin at this distal point that gets further and further away from the upper limit of your meristem, you already put a little bit of a break on the system. So now there is a little bit of elongation zone in the model, but still the meristem is like 3 to 4 times what you would see normally. But then another well-known thing is that auxin has this natural antagonist, cytokinin, and that these two hormones negatively interact with each other. So let's put that in. Didn't work. Because, well, if you put that in, it's basically like, okay, first I help myself making this positive feedback, and now I kill it off again straight away. So what turned out to be essential is that auxin itself actually organizes this cytokinin break, but does so at a distance and with a certain delay. And that helps to first have this startup phase of making a meristem, growing a meristem, and then only at a certain stage put a break on it. And we're now working on the exact logic of how you can do that at a distance and with a delay. So what, if anything, does this have to do with evolution or with animals? Well, So basically what I discussed is summarized here. So we have an aux and platora positive feedback. We have an antagonism at a distance and with a delay, and we have this reflux loop. Well, if we look at animal axial patterning, and what we know most of is from vertebrates, that we know, okay, we have this FGF wind gradient with a maximum in the tail bud, and this is where division and ingression, etc. take place. So that's one similarity. Then it's known that these two positively interact each other. So there's our positive feedback loop. And then it's known that FGF and retinoic acid are important antagonists of one another, and that it appears that once FGF levels taper off but wind not yet, wind starts inducing retinoic acid. So again, it activates its own inhibitor after a certain while at a certain distance. And interestingly, thus far, this antagonism has mostly been implied in regulating this transition from oscillations to pre-patterning so-mite boundaries. But this comparison already gives you also ideas that, okay, it might also be important just for localizing this posterior growth zone. And then finally, another thing for which we can think in that direction is the fact that there is this motility gradient along the posterior mesoderm, and that's similar sort of to the orientation of this auxin transport could play an additional role in keeping this posterior signaling center focused. So in this picture, I'm already hinting a little bit at segmentation. So let's move to segmentation. Well, as my talented PhD student already explained in her talk, I will sort of shortly give a recap of that. So we have only 3 sort of distantly related animal clades that show overt segmentation. Then there's many that have metamerism, and there's also many that have no segmentation. And we can ask a lot of questions about it. And indeed, what many people are interested in, does segmentation evolve? Did it evolve once? Did it evolve twice? Did it evolve 3 times? And we've been addressing these questions with in-silico sort of evolutionary models and testing how easy it is to evolve segmentation. And another question that we can ask, and that was addressed also today with a modeling study, is how does Drosophila type segmentation evolve? How can it evolve from a prior mode of sequential segmentation? But in this context, today, what I'm more interested in answering is why do we get this particular mode of segmentation in most animals, where you have this posterior growth zone, a sort of oscillatory periodic formation of segments. So put simply, what you often see is you have transcription, translation, and then either of the gene itself or some secondary or tertiary gene, in the case of, for example, tribolium, that gives a negative feedback, and that this delayed negative feedback gives rise to oscillations that take place in the bud of the organism. And that these oscillations require a morphogen to keep them going, and then as growth proceeds, they leave this high morphogen level, and the oscillations become transformed into stripes, and you get this sort of space-time plot. So you can ask, how does this evolve? And we've done that in previous study, but you can also ask, why does it evolve? So typically what these models look like is that we have a sort of virtual 2-dimensional world in which in-silico organisms live, move around, and die. To make matters a little bit simple and computationally tractable, because you don't want to, each time you put your simulation on, go on holiday for a month and then come back. We only treat 1-dimensional sort of embryos, and we give them a genome with genes and regulatory binding sites that encode a network. And then by giving them some sort of positional input, the gene and expression dynamics governed by these networks can unfold differently in different cells. And then what we do is we look at gene expression, and we look whether we see some sort of striped pattern forming. And then the more stripes you make, the more segments you have, the fitter you are, and the more reproduction chances you get. And what we did is we gave them a division gene, and when this division gene is on in a cell and reaches a certain level, it can divide, but they can evolve themselves, whether that is expressed all over the place or only at a certain position. And the thing that we tested is, does it matter whether we assume that there's only some transient maternal input to the back of the organism, or that there is a sort of persistent posterior signaling center that we assume has already evolved. And indeed, that matters a lot. So what we found is that if we assume that this posterior signaling center is already present, which is not a crazy assumption. I mean, if we look even outside the bilaterians, it seems that a caudal wind FGF related center has already evolved. Then 9 times out of 10, or even more often, we get a space-time plot like this. So we get terminal addition type growth and oscillatory sequential segmentation. If instead we only provide them with a transient signal, it is apparently in our simulation too difficult to sort of self-organize into something like this again, and then they go for another mode of development. So apparently this posterior signaling center gives a very strong evolutionary drive towards this, this way of making segments. So now let's think a little bit back to our plants, because there we said we had this strong parallel of also having this distal organizer. So are there parallels with plants, or is this completely crazy? Well, here we see a snake, and we see this nice repetitive pattern formed by somatogenesis, and this is an Arabidopsis plant. And we see that under ideal growth conditions there is this nice regular alternation, although here we see an exception, between left-right forming lateral roots, and they're formed at more or less regular distances. So is there a parallel there? I mean, okay, there is like this visual parallel, it's an elongated axis, it's sort of a regular subdivision, but well, it's completely different anatomical structures, so is this crazy? Well, actually what is very intriguing is that, well, we already know that in somatogenesis we have these oscillations, and then we have this antagonism transforming these oscillations into a somite pre-pattern, and that something similar seems to be going on, at least in Arabidopsis. So what has been measured is that close to the root tip there is an oscillation in an auxin reporter. And there's also this auxin cytokinin antagonism going on, and at that point where they intersect, oscillations stop and size are being formed that are competent for forming lateral roots. So this is quite spectacular, right? So people have indeed claimed, also because what they found is that it's not just auxin that is oscillating, but that in fact there are many, many genes oscillating, that, well, there must be something exactly similar going on, that there must also be a sort of root clock, like the somatogenesis clock, with the same kind of gene regulatory architecture behind it. But it hasn't been proven. So let's think a little bit about this. So what do plants want and what do animals want? Well, here we see a poor girl that has a quite severe form of scoliosis, and you can imagine that this brings with it a lot of back pain, and that this will severely hamper her physical abilities to do things. So it is eminent that in somatogenesis what is crucial is that it is precise, that it's reproducible from one individual to the other, and that it's highly symmetric. So if we look at the underlying machinery, then it's very, very clear that the way it is because it wants to, well, want is not a good word, of course, for a biologist, but that it is wanting to do that. So if we look at the oscillatory machinery within a single cell, we see that oscillations occur in different pathways, and we can imagine that this gives us an amount of redundancy that ensures that these oscillations don't easily break down and don't easily change their periodicity. then when we look at neighboring cells, they're talking to each other through delta notch signaling, and it has been shown that this is essential for keeping neighboring cells in phase. So rather than having a sort of salt and pepper pattern of neighboring cells being out of phase, they all go through the same stages, and therefore you can ensure that you have nice, sharp boundaries being formed. And then finally, there are some new insights on how left-right symmetry is being kept, but it's not quite clear exactly how that is working. What we do know is that if you knock out retinoic acid, that then somatogenesis becomes very asymmetric. So you see that there's this whole complex machinery in place to ensure precision, to ensure reproducibility, to ensure symmetry. But then plants. Well, you could say maybe this plant is a poor plant compared to that plant in the sense that it has maybe a less easy life. But for this plant, under its conditions, this is actually the best thing it can do. If it has that much wind and if it has only this crack in the rock with sand in it, it's actually very good to be asymmetric. If it wanted to try to do this, it would probably have failed a long, long time ago. So plants and animals are very different. If this would be a normal condition, the plant will generate more roots if there's less nutrients. On the other hand, if it would have a patchy environment, then it would make more roots, but not in the poor areas, but rather in the areas with lots of nutrients in it. And finally, if it would be a left-right gradient, then it would put all its roots to that side. So, okay, there is this oscillator, but we know that the environmental conditions, they can impact clock speed, they can impact where eventually, on which side the lateral roots will turn up, and it will influence the exact patch of where the lateral roots will be formed. Whereas in an animal, once you have this oscillation, everything else is fixed. So my point being that even though there might be you can consider these lateral roots and these somatogenesis processes as convergent evolutionary outcomes for sort of the similar problem of segmenting a polarly growing elongated axis. This doesn't necessarily extend all the way down to how these oscillations are being generated and translated into pattern. These differences in selection requirements to animals and plants might make it actually quite likely that there are some differences there. And this is something that I would like to address in root models in the next years. So in conclusion, I hope that, that like me, you find these very intriguing convergences between animal and plant patterning that are deserving more attention. And that it's interesting to think about things as convergence and parallelism, not just in terms of what is the eventual outcome, but also in terms of the dynamics of the pre-patterning mechanisms, and even if they don't extend all the way down to all the details. And I hope that I've shown you that with the models you can't just figure out what happens, but you can also get a good insight in why it happens in a particular way, or why it is done in a particular way. So that's it, and I'm happy to take your questions.
Thanks very much for a wonderful talk. So, who would like to ask the first question? Hans. Can he get did you understand it?
Yes, so the question was that in plants, these pin proteins that are on one side of the membrane give this directional transport, and whether I can think of an analogy in animals. Well, not really, because the pins are like across the entire tissue. I mean, of course, when we talk about this left-right asymmetry, then we have cilia going, but that's really more like fluid flow or something like that. Yeah, maybe with some signaling molecules, it's also known that there is sort of a feedback between signaling and then receptor turnover and then signaling again by the next cell, but not, I wouldn't know anything more close. Yeah, they'd rather walk there themselves than maybe, I don't know.
Thanks for a nice talk. I have a question touching on your last slide. So
I'm guessing not this one.
No, no. No, not this one neither. Sorry, this one, yeah. So the kind of simulations you do seem like perfect machines to predict the outcomes of experimental manipulations. And many manipulations, for example, in plants, concern changing the environment. And my question is, are your simulations capable of predicting results for environmental manipulations? And can you hint a little bit on how you would deal with that?
Well, my models are, I'm working on that, but my models are not yet on the scale that they currently model the whole root architecture and then environmental inputs. But for example, well, as I showed, the model can simulate how it responds to gravity, and we've also been making models how it responds to salt. And of course, I mean, a model is only as good as the data that you put into it, so we have to verify whether we are getting the right emergent feedbacks. Because we can model, for example, that if you make a lateral root, it consumes auxin, and it will therefore affect the likelihood that another lateral root arises. But in the back of your mind, we also have to take into account that by doing that, it consumes energy that may also influence the likelihood of forming another lateral root, or that by doing that, it also consumes a nutrient that might impact. So it is quite entangled. So yes, we can do a lot with the models, but it always has to be iterative with experiments, and also between development and physiology in this case.
Are there more questions? Yeah, they're up. Ehab.
Now that I've made her work so hard, you know. So I'm just trying to think about what such a convergence between such divergent organisms would mean. In your opinion, do you think it's because this is the most efficient way for this to happen? So this is a kind of selective ecological argument, or is it, do you think this is the only way for you to build a kind of an axis? Is it constrained otherwise, or is it the mix of both? Of course, but I mean, what is your feeling about this in terms of what this convergence means for evolution?
Yeah, that's a very good question. So I think that, well, the evolutionary simulations were of course only applied with an animal context in the back of our mind, but then it shows that there's multiple ways of doing it. So theoretically speaking, there are more ways of segmenting an axis. So, but what, for example, Renske Vromans, my PhD student, very nicely showed in her work is that often these other modes are less robust or less evolvable. So yes, my money would be on this being both a way that is sort of easy to discover by evolution, because it's much easier to evolve sort of a negative feedback mode than an entire hierarchy, such as, for example, in Drosophila. So it's easier for evolution to hit upon that before hitting upon something much more complicated, and that because of these additional benefits, it's also a better solution because it's more robust. But still, we have to keep in the back of our minds that, well, this has been really focused upon by the Arabidopsis community, but Arabidopsis has this taproot architecture where you have this main root and all these nice lateral roots. But many of our crop species have a main root that at a certain point is not so important anymore, and they make adventitious roots that are higher up. So it remains to be seen whether they use a similar mechanism, or whether, given already that plants have much more variability, yeah, there might also be other processes at work there. So it's not so easy yet to say this.
Okay. Oh, well, the last question, because we have to go to the poster.
Thank you. My question is, how do you challenge your models? Because, for example,
change or challenge? I didn't quite understand.
Like some models, they can be non-falsifiable. For example, you cannot check is it true or not. The model pretty well describes the phenomena, but probably the mechanism is different behind the scene.
Yes, so you're asking, does my model give the right output for the right or for the wrong reasons?
Yes, and also, do you consider this kind of like the logical space of different options and different strategies? And how do you choose the best version?
Yes, well, first of all, I mean, of course, in this talk, I can only show you what kind of a wild type root does under normal growth conditions. But when, for example, building this root model, we also simulate all kinds of mutants, and we also simulate all kinds of auxin perturbation, etc. experiments. So at a certain point, if you have 4 mutants and 10 perturbations simulated and they're all covered, you can sort of safely assume that you're getting the right answer for the right reason, and that chances are that you're getting it for completely wrong reasons are pretty slim. But yes, in designing these models, I guess that's also part of the magic in model making, is that you constantly have to think of, does this make sense, or what would be an alternative? And this is also what I alluded to in being able to do crazy things with models, that you can ask, like, okay, what if instead I still assume that plethora is a readout of auxin? What then changes? And then you can find that it matters. So I think that it is actually one of the great things of models, that you can do that you can try out these alternatives.
Okay, I think that if you want to continue the discussion, that should be during a poster session or at other times. Thanks again very much for this wonderful talk.